Hi everyone, my name is Serene, a first-year graduate student from Biomedical Science program. Today I'm going to share with you on some information and the progress of our research on the discovery of critical virulence factors of Fusobacterium in promoting of oral squamous cell carcinoma. I guess not much of you have ever heard of Fusobacterium nucleatum. This is a gram-negative anaerobic bacteria. That means it might die if exposed to aerobic condition for too long. So we have to grow them inside the anaerobic chamber. They are in spindle-like shape. They look very thin under the microscope as compared to the commonly known rod-shaped bacteria. They are not spore former and motile. So far, they have only been found as commensal flora in the oral cavity of healthy individuals. You might ask, why do we still need to study them? Because there are more and more research show the correlation of these bacteria with different diseases. The number one problem that caused by these bacteria is the periodontal disease. F. nucleatum elongated shape benefits in connecting primary and secondary colonizer to serve as a bridge organism in bioconformation. Beyond the oral health issue, F. nucleatum is also associated with problems across the body, such as the atherosclerosis, inflammatory bowel disease, and adverse pregnancy outcomes. In addition to this, F. nucleatum has also linked to many cancers. For example, the most intensively studied topic, colon cancer, breast cancer, oral cancer, gastric cancer, and pancreatic cancer. And our research will be focused on oral cancer. Oral squamous cell carcinoma, OSCC, is the most common type in oral cancer. In the US, there are roughly 34,000 people diagnosed with OSCC every year. According to the statistic, OSCC accounts for 3% among older cancers in men and 2% in women. And it usually happens on individuals at 50 years old and above. How can we stay away from OSCC? The easiest way is do not smoke or drink. The worst scenario is the combination of these two habits. It will shoot up the risk 100 times in women and 38 times in men. However, some previous studies have also shown the balance of oral microbiota is playing an important role to our oral health. Therefore, the purpose of this study is to identify the molecules of F. nucleatum in promoting OSCC progression. In order to answer this question, the OSCC cell line will be challenged with the absence and presence of F. nucleatum in the cell culture. For direct interaction approach, the bacterial cell and OSCC cell line will have physical contact, while the indirect interaction approach, the cell line will only be treated with bacterial supinated. The effects of the treatments will be observed through this assay, the ability of cell migration and proliferation. Before starting off our actual experiments, we would like to confirm the techniques and protocols that we'll be using in our experiments are at least able to reproduce others' published data. Therefore, HTD116, which is a colorectal cancer cell line, has been chosen as our experimental control. From the cell migration data, the wild-type F. nucleatum strands 23726 and 25586 show statistically significant faster as compared to the untreated group. And the surface adhesive mutant strands, RED and FAB2, show no difference compared to the untreated group. We observe similar outcomes from the cell proliferation assay. By confirming the reproducibility of the techniques and protocols, we are safe to start our experiments on OSCC cell line. With that, I'd like to thank to our collaborators and funding sources, as stated in this slide, and my advisor, Dr. Huang, for the support in making progress of this study. Thank you for your attention. Please stay safe and healthy.